Hello, immunology. All right. So, chapter 6 and 7 are where I think this book really finally gets down to business and gets kind of good. So, we're covering the same things that we've just briefly touched on before, but we're going to do it actually in depth. And this is kind of where all of the rest of the term was leading to. This is where you want to be, and this is where hopefully immunology starts to make a little bit of sense. It's going to take some work, but I think we're going to get there. So let us go. And I'll break this into several parts as usual. So you can watch it in little bite sized pieces. Okay, let's just jump past all of this stuff and let's talk. So yeah, T cells, you know a bit about T cells, right? You got your alpha betas. These are going to be most of the T cells, or what you would consider the normal T cell, and they're going to have a receptor with an alpha and a beta chain. And remember, the beta chain is the big one. That's the one that is the equivalent of the heavy chain on an antibody. And it's going to do more or less the same thing. It's going to recognize an antigen that's going to be presented to it. Then you've got your gamma delta T cells. And gamma delta T cells are their own little creatures. They have their own set of T cell receptors that are made from a gamma and a delta chain. So it's just like a second version of the immune system. And you've also got these invariant T cells, mucosal associated invariant T cells. And these are going to be mostly gamma delta T cells, and they're going to have, the ability to directly respond to bacterial components. And you're going to find these pretty prominently in the gut, if I remember correctly. And I mean, the gut is lined with bacteria that are not necessarily waiting to kill you, but these are things that could very easily get out of hand if they were to make it into the bloodstream, for example. And so, yeah, these gamma delta T cells are just keeping order down there among all that rabble down in your intestines. Okay. Regular standard major histocompatibility complex, your MHCs. And remember, in humans, they're called human leukocyte antigens. But MHC is the general name, and so I think that is fine. You can use them interchangeably because we're going to be focused on the human version of immunology here, which I think is going to be of more interest to you and maybe a little more relevance in what you're doing next. All right, so we got our MHCs and we've got our non-classical. Remember, MHC class 1b molecules. And these are, again, sort of limited diversity, and they're going to present a restricted set of antigens that are very general in type. And so they are like those invariant T cells down in the gut. They're going to be here to generally keep you healthy against really common recurring kinds of threats. Okay, now I'll separate out these two components. So antigen processing is basically chopping up a piece of protein into until it's the right size so it can be loaded onto an MHC molecule. And we have to take it into the cell. We have to get it into the right chamber of the cell. We have to chop it in just the right way. And then we have to get it out of that little chamber of the cell and into the cytoplasm, and if it wasn't already there. And all of that process is the antigen processing process. There we go. It's almost redundant, but not quite, I think. Antigen presentation then is the next step. So once you have processed an antigen, chopped it down to just the right size to fit on an MHC molecule, you're going to load it onto the MHC molecule and then present it to, some kind of cell, probably a T cell. And that is the next step. So together, these are how we show antigens to the immune system. All right, and a brief look at just making sure we're on the same page. These are some of the cells that we're going to be talking about that you've already heard something about. So cytotoxic T cells are going to be CD8 T cells for the most part. That's more or less synonymous. It just means T cells that have T cell receptor on the surface and can kill other cells. So there are a couple classes of CD4 T cells that can do this. Don't worry about those right now. Let's focus on the CD8 T cells. That's going to be the major population there. Although, yeah, as with everything in immunology, there's going to be a case somewhere where the small population of minor things is going to play a big role. And that's just how immunology is. Okay, then you got your effector CD4 T cells. And you're going to have all these different kinds of T cells. So T helper 1, T helper 2, which is going to be heavily directed toward making antibody. T helper 1, you're going to make some antibody, and you're going to do a good job at making other cytotoxic T cells. Th17s, these are another population of T cells that we'll get to a little bit later. And regulatory T cells, or they call them Treg instead of Th sometimes. So those are going to basically shut down other parts of the immune system when they have received signals that say that the danger is over, and you can stop doing an immunological reaction that might actually be hurting you a little bit. To this, I would say we could add, there's a population called Th1 star, which are non-canonical Th1s. So they're going to behave a little differently. They're going to have some markers in common with a Th1, but they're actually kind of their own population. And I don't think the book covers those in any great detail, but these are something that exists and something that people are looking at as potentially being very important in diseases like COVID-19. So there we go, relevant. Okay, cytosol is the cytoplasm. Yeah, just, everything inside the little bag that is the plasma membranes and outside the nucleus, I guess. And vesicular compartments. These are going to be, so a vesicle is like a little bubble. And you can make these things by releasing a little bit of the plasma membrane. You can just sort of snip it off and, join the rest back together. And you'll make a little bubble. It's like a little vesicle floating around. Or you can make these by pulling a little bit of that outer membrane into the cytoplasm and then snipping it off. And then you'll have a little bubble that's floating around inside the cell. We're mostly going to be talking about those. We're going to be talking about vesicles on the inside of cells, and these compartments are going to have names like endosomes, for example, or lysosomes, and yeah, I guess the ER in Golgi would qualify, but mostly we're not going to be talking as much about those as about these guys. Actually, no, we do talk a bit about the ER, so there we go. Fine. Okay. Now this one, let's take a minute to get this right. So you've got a couple different ways of presenting an antigen from one cell to another cell. One way this happens is called direct presentation. And so this is presenting something on an MHC class 1 molecule. And this means the cell was growing a particular protein, which could be a normal protein, or it could be a protein from a coronavirus or from some kind of tuberculosis bacterium, something like that. The cell will shred that up, chop a little bit of that up. Most of it, goes off and does its job, and then it will load it onto an MHC class 1 molecule. And we're going to go through in lovely detail all of the ways that loading MHC class 1 and class 2 molecules happens. And so, this could be just regular, ordinary cells directly presenting to some cell of the immune system, basically showing a cytotoxic T cell that they're making the wrong thing. Or it could also refer to antigen-presenting cells like a dendritic cell. They're able to take in some things from the outside, like things that are floating along in the bloodstream or things that have come out of a cell, and chop them up as if the dendritic cell was actually making this. And then they can show them to the immune system and get a nice, strong T cell response of those cytotoxic T lymphocytes. So think again, mostly CD8 T cells, and definitely T cells that are able to kill cells that they can recognize. Okay, so that's direct presentation, which is you're showing what you're making unless you're like a dendritic cell, in which case you have a special sneaky pathway of showing something that you picked up. But you can show it via class 1 to cytotoxic T lymphocytes. So that's pretty cool. Then you got your cross presentation. And so this is going to be kind of a normal phagocytosis pathway. So your antigen presenting cell is going to eat something, and Did I get this right? Let's see. Let's check. Okay, sorry. Yeah. So direct presentation is exactly what I said. The cell is making this protein, and it shows it. So that's very direct. You see what the cell's making. Cross presentation is the thing that I was getting toward that dendritic cells can do. That's a little bit special. So they're able. This is where you eat that extracellular protein. So eating something extracellular, like an extracellular bacterium or virus or protein or what have you, and then presenting it as if it had come from your cell and presenting it via MHC class 1. That is cross presentation. So they're presenting somebody else's antigen. And direct presentation is presenting your own antigen that you made yourself. So yeah, there we go. Sorry about that. We've got it right now. All right. All right. And yeah, the last thing is going to be cross priming. So CD8 T cells, these are going to be our cytotoxic T cells, are going to be activated by dendritic cells. And remember, dendritic cells are kind of embedded in the tissue, and they're just hanging out and waiting for some kind of antigen to come along, in which case they'll go and present whatever it is they've found to CD8s via MHC class 1 in this case. So we're going to present peptide via MHC class 1, and yeah, this is going to be the result of cross-presentation. So picking something up and presenting it as opposed to direct presentation. So yeah, cross-priming is essentially cross-presentation. Yeah. So, all right, there we go. Fine. Autophagy is self-eating if you break it down. And so this is when the cell will surround and digest chunks of its ER or cytoplasm. Sometimes they'll digest their own mitochondria. Cells do this a little bit just as part of the normal business of staying alive. You know, that's how you get rid of when you have too many mitochondria, for example. And cells will also do this a lot more when they are put under stress. So if you infect a cell with a virus or something, then that is a good way to end up with a lot of autophagy. Okay, and so this is one way. This is a different way that we can take actual proteins from your cytosol, so proteins that you have made, but present them for MHC class 2 molecules. So that's a separate thing. We were all class 1 over here, the direct and cross presentation, and autophagy will let you present on class 2. We're going to go through all these things with pictures, but we wanted to cover the definitions right at the start to get a good little look at this. So yeah, in case you've never seen a cell before, which I don't think is the case, these are the parts. So an autophagosome is when you take something like a little Golgi stack or like a little bit of the ER or like a vesicle, one of these guys, and you just sort of turn it inside out. So it's still a vesicle. It's still like a little circle of membrane. You just stretch it into a big sort of C shape and wrap it around. So an autophagosome is always going to have two membranes, like an outer membrane and an inner membrane. And what happens is that these will go around something like a mitochondrion, for example, and then they will actually join together. And so you'll have the mitochondrion trapped inside a double membrane. And generally the cell is going to digest whatever is trapped in an autophagosome and sort of recycle it. All right. So, yeah. If you got something like a coronavirus, for example, that is going to replicate right in the cytoplasm, then we can actually chop that protein apart in the cytoplasm. We can stick it onto MHC class 1. We can show it to effector or cytotoxic CD8 T cells. And when those CD8 T cells see you presenting something that you should not be presenting, they will then kill the cell that presented it. So yeah. It ends badly for the cell, but it ends very well for the host. And in a multicellular body like we have, we don't want rogue cells going it on their own. You know, no lone wolves. We want everybody being a team player, essentially. Okay, now intravesicular pathogens. So some bacteria like, the one that causes tuberculosis, some parasites like early stage malaria would come in this way. So they'd come in inside of a little bubble. And if this is happening in a macrophage and it happens to get one of these little things inside of it, then the macrophage will chop this up. It'll sort of digest whatever it has just come in, and it will show it via class 2. So MHC class 2, that's something that macrophages are able to show to other parts of the immune system. They can talk specifically to those helper T cells with class 2. So helper T cells are going to interface with your macrophages a lot. And so effector CD4 T cells, and remember the different types? You look back in your notes. All right, see if you get the different types. We covered them maybe 3 slides ago. They all start with TH, like T helper. And so you got your TH1s, your TH2s, and your TH17s, because that's how numbers go in immunology. You've also got your non-canonical TH1s. And so all these are going to get antigen presented to them by class 2. And so that will activate the CD4 T cells and activate the macrophage potentially, actually, yeah, to try to kill whatever it is that is infecting them. So the macrophage may be infected with 50 different bacteria, because bacteria are kind of small and the macrophage is big. That's what the macro part of its name means. And as long as the macrophage can present just one of the parts of one of those invading bacteria, a helper T cell can come along and can program the macrophage to kind of self-destruct anything that's inside of it. The macrophage, I guess it's not self-destruction, it's just getting rid of, the pathogens. Okay. And then extracellular pathogens and toxins. So when a B cell has something that binds to its B cell receptor, like let's say the B cell is specific for coronavirus and a virus comes past, or Ebola virus, or what have you, then the B cell can actually chop up that protein and present it via class 2. And it can show it to effector T cells, and effector T cells can then go and activate B cells to try to fight against whatever it is that has been shown. So, yeah, that's what T cells are there for. They just sort of get other parts of the immune system all riled up and send them out to do some good work. All right, let's check how many more sections. Let's stop at the end of section 6.1. All right, so here's the short version. We're going to go and do the long version in a minute, but the short version is you got something like a dendritic cell, let's say. And so it's going to swallow some kind of bad thing. Let's say it's a fungal spore or something like that, or a little parasite. The dendritic cell is going to digest this thing that it picked up from the outside, and it is going to chop it into little bites. We're going to do antigen processing, and then we're going to do antigen presentation, and this is actually going to involve the ER. We load this MHC class 1 up while it's in the ER, and we're going to put these little peptides, these little parts of whatever it is we just ate on there and show them on the outside of the cell. And typically for this process to work, you're also going to have a second signal, something like a toll-like receptor signal, TLR signal, or a nod-like receptor signal, something like that. And ordinarily, these dendritic cells will just consume stuff and generally not do a very good job of presenting it. But when they consume something and they get one of these danger signals through something like a toll-like receptor, then they will present very efficiently. And so this is aimed at keeping your immune system from over-presenting or presenting normal things even when you're not under any kind of danger. It's basically a way to stop you from having rampant autoimmunity all the time, which I think we can all agree is probably a good idea if we can find a way to do it. Okay, MHC class 2 is going to be a different thing. So MHC class 2, we're going to take things out of autophagosomes, for example, and some other proteins that we brought in on like a B cell receptor, for example. And we are going to shred these things up, and we're going to merge them with a little compartment that has a preloaded MHC class 2. And we're going to merge these two things together. And so then all the little parts are going to lock in. They're going to displace the invariant peptide, which is this little clip up here. And there we go. We're going to have class 2 presenting little bits of whatever the heck we just ate. And it's going to go on the outside. And so we're going to show those to the CD4 T cells for the most part. All right, so that's a good place to pause this first one, and then we'll come back and go through this. And yeah, we're about to talk about the proteasome, and I think you'll like the proteasome. It's a kind of a neat little garbage shredder of a device. All right.